We have developed a technology to produce the next generation of vascular grafts. This new technology platform could fundamentally transform not only vascular grafts for patients with end-stage renal disease, but the entire $1.2 billion market of vascular grafts for patients with any vascular disease. End-stage renal disease occurs as a result of the gradual loss of the kidney function and can become fatal without adequate treatment. The current treatment for end-stage renal disease patients is renal replacement therapy, which includes dialysis and transplantation. However, there's a severe shortage of healthy kidneys available for renal transplantation. Therefore, dialysis serves as the chief mode of renal replacement therapy for the vast majority of patients until a kidney transplant becomes available. A functioning vascular access is necessary for hemodialysis. Currently, arteriovenous PTFE dialysis grafts are the most common form of hemodialysis vascular access in the United States because they are relatively easy to apply and available off the shelf. However, they have extremely high failure rates due to thrombosis, stenosis, and infection. A salvage procedure to maintain graft patency, such as thrombectomy, angioplasty, or surgical revision, would be required in 77% of the patients at 1 year and 96% at 2 years, which means the primary graft survival is only 23% at 1 year and 4% at 2 years. These dismal outcomes have a high associated cost, significant to both patients and the healthcare system. In the first year of hemodialysis, as many as 25% of all hospitalizations and as much as 50% of all healthcare costs can be attributed to care related to maintaining vascular access. Vascular access dysfunction is a central cause of morbidity and hospitalization in the hemodialysis population in the US and costs the healthcare system approximately $1.7 billion a year. Synthetic PTFE materials possess no regenerative or growth potential, and one of the major causes of their high failure rates is the lack of a functional endothelium. So what is endothelium and why is it so important? The endothelium is the inner lining of native blood vessels and plays critical roles in maintaining normal vascular functions, such as antiplatelet adhesion, anti-inflammatory responses, and modulating the interactions between the blood components and the surface of the implanted vascular grafts. Establishment and maintenance of a healthy endothelium is crucial for the prevention of clotting and narrowing of blood vessels. Unfortunately, limited progress has been made in this field for the last 5 decades. Therefore, there's a significant unmet clinical need for vascular grafts that are able to maintain superior functionality and remain open to persist after implantation. This is where our technology comes in. Our innovative coating technology specifically targets endothelial cells and endothelial progenitor cells, which addresses this problem right head on. How did we come up with this novel technology? 8 years ago, after I finished my postdoctoral training at the UC Berkeley Bioengineering and Stem Cell program, I joined the UC Davis faculty in the Department of Surgery. Being a PhD researcher in the clinical department, I have the privilege to work very closely with my surgeon colleagues, including those who are taking care of hemodialysis patients. I knew we needed a different and better strategy. And that's where I saw the opportunity. I started collaborating with Dr. Kit Lam, a very well-known cancer researcher and chemist on campus, who invented a high-throughput screening system called one-bead, one compound, which has been successfully applied in cancer drug development. The question was, instead of looking for drugs that can kill cancer cells, can we use one-bead, one compound to identify molecules that can support endothelial cell growth? The answer is yes. We identified a specific molecule that possesses strong, stable, and specific endothelial cell targeting and supporting function. This is the workhorse of this coating technology. Our coating technology does not change the physical properties of PTFE grafts. As you can see here, the conventional PTFE graft and the coated graft, they look identical. There's no difference that you can tell from the outside of the graft, and the surgical application of the coated grafts is no different from the conventional grafts. However, from the inside of the graft, there's a significant difference on the molecular level. Conventional PTFE grafts do not have any specific regenerative functions, and once implanted, various types of cells such as red blood cells, inflammatory cells, and platelets from circulation could randomly attach to the graft surface, causing the blood clot formation in the short term and the narrowing of the graft in the long term. In contrast, PTFE grafts coated with our technology are empowered with these ligand molecules. These molecules function like sticky hands, similar to this kid's toy. These sticky hand molecules coated on the luminal surface of the graft can specifically capture only endothelial cells and endothelial progenitor cells from circulation. There are hundreds and thousands of these sticky hand molecules on the luminal surface of the grafts that support the capture and promote the growth of endothelial cells, and essentially recreate a functional living endothelium on the synthetic vascular graft. We have carried out preliminary efficacy studies in both small and large animal models. We first tested our technology in the rat carotid artery bypass model using a very small diameter vascular grafts. As you can see here, these very small diameter vascular grafts became clogged easily after implantation. In contrast, our technology-coated grafts showed a significantly higher patency rate than the untreated grafts. 6 weeks post-transplantation, 5 out of 6 treated grafts were patent versus only 1 out of 6 untreated grafts were patent. To assess the feasibility of our technology in promoting the functionality of clinical-grade PTFE grafts, we further conducted preliminary efficacy studies in the large animal model. 4 weeks after implantation, we retrieved and longitudinally cut open the graft, and we found apparent blood clots on the luminal surface of the untreated PTFE grafts. Under high-resolution scanning electrical microscopy, we can clearly see significant red blood cell attachment and platelet aggregation. In contrast, on the luminal surface of the PTFE grafts treated with our technology, we do not see thrombosis, red blood cell adhesion, or platelet aggregation. Rather, we can see a smooth layer of endothelial coverage on the luminal surface of the grafts. This novel technology has a significant impact on transforming the functions of a conventional vascular grafts. Vascular grafts coated with our technology mimic the body's native vascular system, regenerate of a living endothelium on the luminal surface of the grafts, essentially creating an organic vein which provides resistance to complications associated with the standard of care grafts. There's a clear, well-developed market for vascular graft and dialysis. The global dialysis market is expected to reach $108.5 billion by 2025. We don't expect healthcare coverage of the PTFE grafts to shift significantly with the addition of our coating technology. The price of the graft itself may change when the ligand is added, but this can be justified by the subsequent lowering in healthcare costs from decreasing repeated hospitalizations. We have already filed PCT patent applications with worldwide coverage. Our patent claims are not only limited to hemodialysis vascular grafts, but also cover the application of this technology to a wide range of intravascular devices, including stents, cardiac valves, and catheters, where a living endothelium is needed. We have assembled a strong team to move this technology forward. I'm a co-founder and the co-inventor of the technology. I have over 15 years of experience in bioengineering and vascular device development. Co-founder and co-inventor of this technology, Dr. Kit Lam, is a physician scientist and a well-established investigator in the fields of chemical biology, chemistry, and nanotechnology. He will lead the technology development, chemistry optimization, and prototype production. Co-founder Dr. Elisa Panich is a serial entrepreneur. She successfully founded 3 startup companies, and she has extensive experience in company management, regulatory path, and FDA interaction, and will primarily direct the development of the technology to gain FDA approval. We also have several well-regarded surgeon colleagues who are serving as scientific and medical advisors for our team. Overall, our team possesses a unique multidisciplinary view and has a long-standing proven experience in drug and medical device development, commercialization, and clinical use. So what's next? Currently, we're seeking strategic partnerships and investments as we move our technology to market. We're seeking about $12 million to allow us to refine the prototype, select a manufacturer, complete the large animal studies, and obtain regulatory approval. Application of our technology would not only financially benefit the healthcare system in saving resources, but also having a significant positive impact on patient outcomes. For patients on hemodialysis, fewer reinterventions will certainly prove the most impactful towards improving patient quality of life and reducing the risk of repeated re-exposure to reintervention procedures. While our initial plan is to make our technology the standard of care vascular grafts for hemodialysis patients, this coating technology can be used on stent, catheters, chemoports, virtually on any device or surface that comes in contact with blood. Thank you very much for your attention.